We're thrilled to welcome Sri Kosuri, co-founder and CEO of Octant, to the show today. Thank you once again, Sri, for joining us.
Thanks for having me.
Let's kick things off. Sri, could you share a brief personal intro?
Sure, yeah. Prior to Octant, I guess I was a professor at UCLA. My lab worked at the intersections of large-scale approaches to understanding biology using functional genomics, synthetic biology, computation. I guess prior to that, I was in and out of startups and academia over the years. I helped start a company, had a flagship called Joule Unlimited, which was a algae to biofuel company. And then I spent about 5 years working with George Church and the Wyss Institute as a staff scientist there. So kind of been in and out of the industry over the years.
Interestingly enough, I had a friend or two who worked at Joule, so I had no idea that there was a connection there.
Yeah, I was officially the first employee of the company, I guess.
Oh, quite incredible. It sounds like you've had a long history in the entrepreneurial space. Before diving too deep into Octant, can you just provide a brief overview for us?
Yeah, Octant is a small molecule drug discovery company, but we use basically high throughput synthetic biology and synthetic chemistry approaches, combining that with functional genomics to kind of empirically explore these spaces at incredible scale. And we really bring these technologies together to build small molecules that modulate multiple targets, multiple pathways, or multiple mutations. So these kind of multi-targeted small molecules. And we apply them in disease areas where we think such approaches are necessary. That's basically the fundamental approach that we take at Octant.
Very cool. And I'm excited to dive in and learn a little bit more as we get into specifics. Michael, over to you.
Awesome. So thank you for the introduction. In 2020, Octant emerged from stealth with a mission to develop treatments for complex diseases using the technologies which you mentioned across synthetic biology, functional genomics, next-gen sequencing, and computation. So could you share more about the founding of Octant?
Yeah, you know, I was a professor at UCLA. I had no real intention to ever leave. I was having a blast. It was a great time. The lab was great. But I think it was just one of these things where maybe 5 years ago, or maybe a few years before we started Octant, many of the technologies I had been working on in my career, so like DNA synthesis, cell engineering, genome engineering, functional genomics, bringing that together with computation, they all started coming together in these kind of magical ways where an individual in my lab, without really any equipment, could build these massive experiments through the use of molecular barcoding technologies that can be read by sequencing. And so all of that was starting to come together, and this ability to explore biology at scale was really new. And so it just seemed like there were opportunities arising. And I think there were others bringing those approaches together in the same ways, but a lot of them were really focused on novel modalities, whether that be like gene editing, AAV, antibodies, but not really bringing them together in the same way to tackle complexity in biology and disease. And it just felt like a very large opportunity. I think maybe the second thing were the people. Ramzi Homsani, my co-founder, I've known for a very long time through family, and he was thinking about his next thing. He had very kind of senior informative roles at the early days of Google for a long time, and then Dropbox. He was a bioengineer as an undergrad and was thinking about coming back. And he's a great operator, and like I'm good at lots of things, but bad at lots of things too. And I think we were just really complementary, and having been in and out of startups, like it's not just about the science. And so as the two of us were like, let's try to do something together. And I think that's kind of really the genesis, and that really started in earnest, when I was shutting down my lab. I spent about 2 years making sure everyone in the lab had places to go, and we could like exit gracefully. And then we kind of I moved personally, and then the company as well in 2019 to move up to the Bay Area. And that's kind of when we really got started.
That's exciting. It sounded like right ideas at the right time, and surely a large leap to take after getting such a great position at UCLA. So we're excited to chat a bit more about the technology that you're working on. So before we dig in too deep, could you provide a bit more context for the audience about some of the challenges in drug development particularly within complex disease, that have been limiting progress in the past and the future that you're trying to go after at Octant?
Yeah, I mean, so much of my research also in those days were focused on neuropsychiatric, well, in a bunch of different areas, but in complex diseases in particular. But I think basically most diseases, the problem in drug discovery isn't like, you know, finding a drug towards a target we all know about and we know what to do, it's just we don't know how to do it. There's some cases like that. But I think in most cases, it's really about figuring out this massive complexity in disease biology and knowing what problems to solve in the first place, right? So if you imagine a disease like depression, you know, at the molecular level, like, is it one disease? Is it hundreds of diseases that manifest in the same way? I don't think we have good answers to that question. And even when we have like a great target to go after in a disease, you know, a particular target or gene might have many functions, and figuring out like what fundamental mechanisms are leading to the dysfunction is important, because ultimately that's what you need to go drug, right? Like, and so that uncertainty, I think, is what drives most of the challenges in drug discovery. Like, if we could pick the right target or the right mechanism of the right sets of targets, you know, drug discovery would get a lot easier. I think that's the challenging part. And a lot of what we try to do at Octant is like try to find better approaches to do that. And then, yeah.
Yeah, so can you share a bit more about some of the approaches that you're working on and how you hope to address this?
Yeah, you know, I think one of the exciting things that's happened over the last decade is really this accumulation of human data sets around genetics and drugs and the environment and how on people and how they like don't impact traits and diseases, right? And that's fundamentally, I think, game changing in a lot of ways. But one of, you know, ultimately the hope is that we can use those data to really use humans as the model, right? Whether that's our genetic diaspora or experiential experiences and lifestyle diaspora, and use that to uncover the complexity in disease. What we try to do at Octant is to build the data sets that can help us get at the causal connections between those things. And what that means is like, you know, ultimately the way that genetics, drugs, and the environment go and impact traits and diseases is still through altering cellular and molecular physiology. And can we build the data sets to understand how those relate? And so we use our platform to build some of that understanding, and then also use the other half of the platform for then go build drugs towards those molecular mechanisms that we find to be causal. So that's kind of one of the big approaches that we take to try to de-risk some of that uncertainty.
Can you explain a bit more about the platform, perhaps at like a higher level, and then we'll dig into kind of each of the separate parts of it after?
Yeah, so I mean, we have two main kind of strategies we're running on our platform these days. Maybe by explaining one of them, it'll become a little bit more clear. But you know, so we have a program in like common diseases and obesity, and another set of programs in rare diseases. Happy to talk to about either, but maybe starting with the rare disease side, because I think it's simpler to understand. If you think of a rare disease genetically is usually you're losing the function of a particular gene, one of your 20,000 genes, right? And that's like kind of, if you remember biology and Punnett squares, they're like, it's kind of Mendelian diseases. They're single mutations often that cause the problem. So you're knocking out a gene's function. Interestingly, there's lots of ways mutations can knock out a gene's function, right? You can delete the whole gene, you can put what's called a stop codon in it and stop making the gene. But one of the big ways is something called missense variation, where like you change an amino acid and something happens to the protein. And one of the big things you can do to a protein is slightly misfold it, right? And the types of proteins that are most sensitive to that are proteins that are trafficked throughout the cell. So they go through this thing called the secretory pathway. And lots of rare diseases are caused by mutations that get the protein stuck in that pathway. Like they just don't get to the right place. The proteins are otherwise fine, they just don't get to the right place. So the classic example here is cystic fibrosis. There's one variant that dominates European populations. that like 85% of people with cystic fibrosis of European ancestry have the same variant, right? So a company called Vertex Pharmaceuticals built a drug, that rescues that protein by like building a small molecule, providing more like stability in where it gets stuck in the ER of the cell, and bringing it to the surface and allowing it to function a little bit better. And they've turned that into a whole franchise of molecules. And so these are called correctors or chaperones. So that's super interesting, right? Like all of us, you can build a small molecule to rescue an individual mutation. But what they found over the years is actually you can rescue lots of mutations in that long tail of the 15%, but only ones that share that same kind of form of mechanistic dysfunction, like getting stuck in the ER, So what we do at Octant is like, can we turn a lot of other rare diseases into something that looks like cystic fibrosis, both commercially and in the clinic, right? So meaning what we do is in lots of other rare diseases, there's not like one dominant mutation often, right? There's lots of different mutations. Some of them, just kill the protein's active site, say, but many of them cause this other problem, and it's hard to know that ahead of time. And so what we can do is build every possible variant that might ever exist in the human population into these human cell line models, and then measure all these different things, like where is it going? Like, is it still functioning? Can it be rescued, right? So like, those are the types of things we can measure when we try to split these things into like mechanistically related. In particular, we're looking for trafficking mutations, subsets of mutations. And so what we do then, after we do that, is then go try to build a small molecule towards those large subsets in ways to lop off a very big part of the patient cohort, like, 30, 50, 70% of the people that suffer from that disease. And so those are the types of things that we try to do at Octant in that program. Is like, and the first thing we're starting there is in autosomal dominant retinitis pigmentosa, where a gene called rhodopsin misfolds and gets stuck in the ER. So these are the types of problems we work on, but we think there's like 20 or 30 diseases that have the same mechanism, and we could, if we can show this here, we can just kind of start turning the crank and building small molecules against these kind of large subsets.
That's an extremely interesting topic, being able to kind of convert, in some sense, part way from a misfolded or in some sense improperly folded protein to a format where we know what to do with, and there really must be a huge library of various protein permutations or misfolded permutations. We're curious, could you share more about the target identification process for how you find these proteins and identify them as potentially treatable?
Yeah, so in that particular set of processes, I mean, in the correctors, it's actually fairly straightforward, right? So in the sense of you have to look for diseases where, the patient population isn't dominated by things called like stop codons. Like, it's not mostly due to like the protein not being there, it's mostly due to these kind of missense variants. That's like one clear indicator. Like a second clear indicator is like, is the protein a membrane protein or trafficked or exported or goes to the lysosome? Like, does it go somewhere in the cell that it needs to be? It's another clear indicator. And then third is like, actually just, is there evidence that trafficking is playing a role? And then we can go further and then start building these data sets around like what percentage of the patient population like is mistrafficked, right? Like, is that a significant portion? So then we can start building our own data sets that correlate with these other ones and try to understand like biochemically what percentage of this mechanism is driving in. Is this worthwhile for us to go after from a commercial and clinical standpoint? So like those are like, that's one big area. In the case, we can talk about the other cases, but oftentimes what we're trying to do is build our own data sets that kind of let us, whether that be with genetics, or sometimes it's just like old sets of drugs and clinical trials and trying to see them across different pathways and trying to understand the differences and how that relates to phenotype. So usually we're trying to build our own data sets that give us some insight into these kinds of problems, and then like let us go after them using the platform.
Can you maybe share a bit more about the platform that you're using to build the data sets? I suppose what would make it different and more advantageous versus other companies which may be interested in exploring rare disease space?
Yeah, so like I think it's a combination of things, but if you just think about from the technical standpoint, like we're really good at this process called like deep scanning mutagenesis, and what that means is what we do is make the mutation of a particular receptor, but we do it lots and lots of times. And you can imagine doing that like in a big cell factory where each well does each one of these things. But the way we do it is like a class of kind of experiments called multiplexing. And all that means is we engineer a cell line, it has the right mutation, it has a reporter that we want to use for looking whatever particular function, say in this case, trafficking or getting stuck somewhere. We can use synthetic biology to like build those circuits to like measure those things. And then what we do is if it is stuck in the ER, it spits out a barcode. Basically, it produces from DNA to RNA this a barcoded reporter. And all that barcode is just a series of, you know, ATCG or GCTA. And the point of that barcode is like the level of the barcode gives you like some readout on that particular function. But what it lets you do is that the next cell right next to it might have a different variant and a different barcode, right? And so in a single dish, you have hundreds of millions of cells. And all of a sudden you can do hundreds of millions of experiments if you can get all of that process right. And so like one person can just sit there and add like, you know, do this whole thing and get the whole data set at once. Of course, this includes a lot of like engineering and computation and all these things, but ultimately it's not a factory that's doing this, it's one dish. And it's this ability to leverage the fact that you have large numbers of cells and each one's a different experiment, you can do it. So that's one of the big ways we do that. Another big kind of thing is once we've identified, say, the subsets of variants we want to go after, we have this ability to, like in a single well of these kind of high throughput format plates, run tens of thousands of experiments where in each well you're doing like, say, 100 or so different, say, variants or different targets or pathways or like kind of counter screens. And then we can do like high throughput chemical screens on top of that. And that's how we build the drugs, right? So then we can like, we have a high throughput synthetic chemistry capability that lets us alter kind of chemicals in more subtle ways to basically optimize on these kind of multimodal readouts. And so that's why we think in general, we tend to be pretty good at these kind of multi-parameter optimizations. And so we try to find problems where that's enabling.
Yeah, that's interesting. So let's say then you have a list of potentially addressable targets. So then can you explain in the case of rare diseases, how you would shift into the drug development or drug discovery process after you've identified the targets?
Yeah, so in that case of like the autosomal dominant retinitis pigmentosa, we have a suite of mutations that we want to correct. There we can kind of look at like, are they corrected, right? So we can build functional reporters for all of those things and then test all the mutations. And then what you can do is then do high throughput chemical screens on top of that. And our chemistries are really focused around this idea that like, you know, it's not hard to get like an initial hit or something like that. The hard part is then just like really optimizing that. And so what our chemistry does is not to build all of chemical space, but really start in a local area of basically a core of a molecule. And then we can add like, you know, 5,000 fragments here, or 5,000 fragments there. And we look at how that alters the ability of these molecules to like work against those multimodal readouts. Does that make sense?
Yeah, we'd be interested now in pivoting a bit to the complex disease case as well. So we'd love to hear if you could follow from perhaps like the target discovery process and then go through the drug development process as well.
Yeah, so one of our other big areas is in engineering kind of small molecules that hit multiple targets and pathways and not hit other targets and pathways. And we focus this in an area, in a class of targets called G protein coupled receptors, or GPCRs. GPCRs are a large class of receptors, the largest class of receptors. There's about 800 of them. Half are like measure, you know, like responsible for your sense of smell and taste and sight to some extent. And the other half are like controllers of cell physiology, right? So they're great drug targets. So I think it's on order about half of all prescriptions target these receptors, about a third of FDA approved drugs. And so they're just central drug targets. They've been known for a long time. But the interesting thing about these kinds of receptors is a lot of these drugs were found by accident, like atypical antipsychotics initially, and then like small changes to those accidents right over time. And they tend to hit many receptors at once. And each receptor can turn on multiple pathways, and these chemicals can modulate the extent to which those different pathways are modulated. And so this is so the first thing is called polypharmacology. The second thing is called functional selectivity or bias. What like the basic bet we're saying there is like, look, like the industry as a whole has been like really focused on specificity here. But even in the cases where we achieve that, like that always doesn't lead to a better drug. And so I think broadly, both endogenous agonists and natural products and all of these, like all the illicit drugs you've ever heard of, right? Like they all hit these receptors, but they hit them as almost chords rather than individual nodes. And so then our job becomes at Octant is really, our approach is really like, can we figure out the notes in the chord that we have to hit to alter the phenotypes we want and not achieve like the kind of toxicities that are not there, right? Or that we don't want. And like once we identify the notes in the chord, like how do we engineer small molecules to hit the notes in the chord? And so we started that in a class of receptors called the melanocortins. Melanocortin 4 of the 5 receptors is like the largest cause of Mendelian inherited obesity. Many drug programs have gone after this. Most have failed due to on-target toxicities. And so what we try to do is like look at all those failed programs of some that worked and like some of them are peptides. Like how do those relate to like all the different pathways that can come off of there and all the different receptors? Like what are their molecular fingerprints? How does that relate to phenotype? We can also use human genetics there and build these kind of deep scanning mutagenesis to understand which pathways relate to things like BMI and or like off-target effects. And so we develop our hypotheses around what the sets of notes are. And then we plug in our high throughput synthesis and chemistry capabilities in order to like say like decompose some of these past attempts and recompose them in the ways that we want. But with these particular notes in mind. And so that's kind of another, and then if we can do it here, then there's many other classes to go after, like the serotonin receptors, the dopamine receptors, the adrenaline receptors, they're all like this, right? And so those kinds of problems have been traditionally hard to approach. And like, we think we can take big approaches there, starting smaller in a smaller class of receptors, but over time, it's the same kind of thing. Like, how do we build small molecules that hit the right set of things that we want? And so it's the same kind of multi-parameter optimization that we kind of run the same platform on. We're just measuring different things. So rather than measuring, like trafficking of a particular receptor, we can measure like calcium like signaling or cyclic AMP signaling or the different types of signaling that GPCRs turn on.
Yeah, it sounds like a very, very large multi-parameter optimization problem. We're curious, so for a lot of these different complex diseases, in addition to there being certain receptors which may be involved in multiple different pathways in the body, there are lots of times there may be patients with a variety of different etiologies. Yeah. And so I'm curious if you're aiming to establish, say, like a panel of targets for all patients in a given disease, or if you're looking to perhaps like segment patients based on commonalities in say this set of a variety of targets for a given group.
Yeah, so in our correctors program, that's like super straightforward, right? Like we're saying, we're trying to build drugs towards like these 500 mutations and we can't touch these other 1,500, but these 500 mutations constitute like, whatever, a third or half of the population, but like we can target them and we know that we can go after those. And so the trials get put that way, the sequencing, so like that is exactly what we do. In the complex disease space, what we're not doing that, right? Like one of the reasons to go after complex diseases is they're very common, right? There's very large subsets, but what we're not doing in that particular program is, what we're saying is like, we're using the genetics and the differences to kind of really understand like what things to modify in order to build a safer drug for the general population. So I think towards a neuropsychiatric disease problem, for example, when it's like depression or like, that's a much bigger problem. Like, I don't think Octant as the company we are right now probably can't go tackle that problem, right? Like, I think it'll be really interesting on how that goes. Like, are there going to be biomarkers or are there going to be genetics that really separate these things? I don't think anyone knows the answer to that yet. And we certainly don't, right? So, but I do think these types of approaches are likely to be helpful in thinking about that.
Yeah, absolutely. And was just curious. I know it's a super huge challenge and very tough to navigate through.
Yeah.
And kind of going off of that as well, you know, having identified, let's say, like a list of targets that may need to be modulated together or to varying degrees, do you foresee treatments which say there are multiple different compounds or molecules that an individual is taking as part of one treatment? Or are you envisioning a world of, say, like one type of molecule which can modulate different pathways in the body?
Yeah, so I think this is often a point of confusion. But what we're saying, we're not saying we're going to take any two random targets and build a drug that modulates both of them. What we're saying is that a lot of these targets exist in families. So like the aminergic class of receptors is a set of 35 receptors. They bind serotonin, adrenaline, acetylcholine, histamine. Most drugs that target, like most atypical antipsychotics, will hit 15, 20 of these receptors because all these molecules look alike that they're trying to sense. And so the drugs look somewhat like them. And so they all, like the receptors have a lot of what's called polyform. So a drug, so this happens in GPCRs, it happens in kinases, it happens in like lots of receptors like this, right? Because they're trying to sense the same thing endogenously, you're trying to like fool it a little, like fool one of the receptors into thinking it's like adrenaline, but you know, the other receptor is like, oh, this is not adrenaline, right? So like, there's like a lot of that goes on, and that's just a hard problem to solve in general. We think we're good at solving that. But what we're not saying is that we're going to take two random targets. So we're saying in these spaces where there's already a lot of polypharmacology and bias, like how do we be more rational about engineering that in the right set of ways? So that's kind of our approaches. We're not doing two random targets in that sense. So that's how we think about it. In terms of your question on like, are combinations really interesting? Yes, like there are some really interesting approaches. Octant is not taking them yet, but I do think it portends a future. Like there's a company called Karuna that I think is really, where there's a set of, this is in the same class of aminergic receptors, but like, there's a drug that's super, I forget exactly, it might be, I don't know if it's major depression. I don't remember exactly what the indication, it's some neuropsychiatric indication, but the drug works, but it's super toxic to the peripheral nervous system, but the actual working part is in your brain. And so what they also do is add an inhibitor at the same time. So it's a fixed dose combination that doesn't get into the brain, right? So you have an activator and an inhibitor in your body, but only the activator is in your brain. And so you can get over some of that same kinds of problems that using this kind of dual approach. And I think more and more you'll see more and more approaches like that, right? Because I think the fundamental point is like, I think all of these companies, us, many others, there's no magical silver bullet target out there. Like biology is not built, it would be a rare disease if it were such, and so how do you get over the fact that biology uses all these components in different ways and different things? And how do you engineer new types of therapies in that world? And so I think those general types of problem is one that we're trying to go after, many others, and using different approaches, including combinations. Yeah.
I'd love to double click on that for a moment when you were talking about targeting numerous potential biological targets within the same class, within the same family. And just because, and forgive me, maybe this is misunderstanding what you were saying, or just my current limited understanding of the biology. Part of the reason pharma goes after specificity is because hitting a combination of those targets simultaneously can lead to pretty significant off-target effects. So are you trying to go after subsets within the family that lead to synergistic combinations? That would be my guess. Or are you concerned about that sort of off-target play?
So yeah, so like
specified multi-asset combination.
Yeah, so I think generally, I think that was a long time, like the basic idea, right? Which is, oh, we're going to make a super specific drug to the one thing that we want to hit. And we just need to identify what that one thing is. And so, people have done that. What you often find is either that drug isn't as efficacious or it's actually more toxic. And so like toxicity arises from off-target toxicity, but actually one of the most pernicious things is on-target toxicity. It's usually like that same target in your, this is the example of Karuna, like the same target in your body, like in the periphery, is like, it's bad there, but in the brain, it's like gangbusters, right? So like, how do you solve problems like that, right? So like just pick a different target, but like it's a really great target, right? So like how do you deal with those kinds of problems? Like, one of the ideas around polypharmacology and bias is that like lots of diseases, it's like a particular cell type is misbehaving, like beta cells in diabetes, or like in the brain, a particular type of neuron. Each of those cells has dozens of receptors, dozens of GPCRs that communicate through the same pathway. So one of the general thoughts in this field is like, what's happening with polypharmacology and bias is this ability for drugs to hit, to synergize on a particular cell type or maybe synapse or pathway that might be interesting. And that's how like natural endogenous agonists often work too. And so you're basically trying to play the right set of notes in that particular and not hit the wrong ones. But it is usually often this ability, you can almost achieve less toxicity by hitting multiple receptors, if that makes sense.
That is pretty interesting. That
So,
sounds like, as you're saying, that level of modulation in the where and the what you hit.
yeah, and so the
might be
hard
able
part
to-
is like, what are the notes that you want to really play? And like, that is definitely hard, right? And there's different ways to think about that, like single cell sequencing is starting to reveal repertoires, or lots of really interesting tools like chemogenetic tools where you can kind of use that as a guidepost. But there's also just like taking old drugs and their failures and the past approaches and looking at the phenotypes and seeing how they differ in order to make those things, or you can actually use human genetics. So there's a lot of new data that helps us guide a lot of those kinds of questions.
And it sounds like you're collecting a lot of it yourself through your.
And then we can collect a lot of it ourselves. So like
Yeah.
we took, in our particular program, we took all like hundreds of compounds and built them and then, run them against these assays in order to be able to like map out like how the differences in polypharmacology and functional selectivity relate to phenotype, or mutations, and how mutations lead to changes in BMI in this class of receptors. So those are the kinds of things that we can do well.
And what led you to choose some of the indication spaces you're tackling? Going
Yeah,
from rare diseases to obesity, there's a lot just there.
Yeah, so I think already that's a lot, right? So I think early on, we started the obesity thing in 2020 when we just launched. It was just a really good platform fit. Like a lot of like we included, thinking a little bit about like obesity is getting really interesting. As you might have seen, like there's been a bunch of big successes in treatment of obesity as of late. Tirzepatide being the latest one a few weeks ago. Like, the standard of care in obesity used to be you lose 3 to 5% of your body weight. That was like, and mostly uppers, right? Like, and then it went to 10% last year and then 20% this year, right? So so really big progress, a lot more excitement in the field that we actually, after like 50 years of lack of excitement. So we started getting excited about that space too. And just generally the things that are working are these kind of multi-targeted GPCR things. And so like, there was just reasons to think that, and all of those things are injectables. Could we build small molecules in the same way towards them? And so that was one of the big things that we were thinking about there. I think it's hard. Like, I think there's also challenges with that. Like we, as a small company, probably couldn't run like large scale obesity trials ourselves. So what does that mean? I think the rare disease thing actually came more organically. It turns out the melanocortin-4 receptor is also a rare disease. Like it's the largest cause of Mendelian inherited obesity. And what we started seeing is the small molecules we were creating actually rescue large subsets of those variants, because we had built all the possible variants of this receptor into the, and so that just got us thinking about this idea of like, how many rare diseases are caused by mutations that lead to mistrafficking? Like, could we start? So I think that grew out organically and we got very excited about that. I'd say a lot of the kind of future pushing is really, we think we can make pretty large impact in really severe diseases that have no other standard of care, and we can push those a lot quicker in some ways. So, and do more of them. So, I think like any platform-based biotech, you're making a portfolio of risks and decisions and market size and like what's possible and what's not. And so we think a lot about like, how are we taking differentiated bets that kind of mitigate as much risk as possible? Some of them biological, some of them commercial, some of them clinical, and we try to build a portfolio of things.
And as you do so, it's clear that not only are you pioneering an exciting platform to address the drug development challenges in complex disease, it's also clear you've overcome a number of pretty significant challenges as well. Could care to share more about what some of those might have been and what to overcome them?
Yeah, I mean, there's been a lot, like, I think anyone will tell you starting a startup is hard. It's fun, it's exciting, but it's always like something's always on fire, right? Like, you know, we moved here at the beginning of like basically beginning of 2020, end of 2019, pandemic hit. We were trying to build a chemistry platform during that time. You know, we actually open sourced the back end of our platform and released a COVID test based on, because we can basically measure RNA and like at very high throughput for cheap. We open source that, we kind of ship that to a bunch of places. So you just never know in those early days, and we were still pretty small, like, you know, I think 20 people late in 2020. And, you know, as we've built, I think it's just learning along the way, building the right team, like getting the culture together. I think all of those things are hard. I think from a challenges standpoint, I think, you know, thinking about commercial and clinical and like how long it takes to get there, what can we capitalize ourselves. I think most startup founders kind of go through that and try to understand that better and better. But I think overall, like we've done a lot. So like, it's like, it always seems like challenging in the moment. And then like you take a step back and you're like, oh, wow, look at all the things that we built.
And it's clear you're going to continue building. I'd love to take a pause and talk about that and think something you said earlier that I really appreciated was making sure that as you closed down your lab, everyone landed on their feet. And so thinking about that, it's clear you care a lot about the people, the team, the culture. I would just be curious, given the complexity of the spaces you're tackling, how do you think about building the team at Octant, which I'm sure you'd probably say is one of the greatest accomplishments of the company so far?
Yeah, you know, I think you know, building culture is a really interesting kind of exercise. I wrote a blog post about this recently, and like one of the biggest learnings was like, whatever cultural things that you want to build into your culture have consequences. And that everyone might or might not like, right? But like, but really what defines your culture is what you decide to do. So for example, you know, one of the things with people that we've always tried to hire is this term we call, like not us, there was this essay by Sean Eddy a long time ago called anti-disciplinary science. It's like ANT, it's like before discipline. And I think the basic point of the article is like, look, own problems, and you want interdisciplinary people, not interdisciplinary teams. You want people that like, you know, may do experiments and computation. You want people that can like understand each other's problems because they've been there and they've done that. And like, I think the last, you know, 5 to 10 years has seen a new breed of scientists get trained that way. And so we definitely try to hire those types of people. And it's been a big, I think, advantage of Octant. So like, do we have the best machine learning engineers in the world? Probably not. Do we have the best like obesity scientists in the world? Probably not. Do we have by building these kinds of people that can think across these roles and work across these roles, it lets us take differentiated bets into the world, right? We're not doing what we're, you know, in some ways we're not the 35th, you know, CRISPR company going after sickle cell. And so like, we are taking kind of bets into the world in therapeutic areas that I think are really important that like no one else seems to be working on, right? And it's because of this ability to, I think, kind of build, like hire people that can talk across those boundaries and work, and not just talk about, right? Like actually work across those boundaries. And that's been a big, like almost central thesis in building the company. I think culturally, we're also a little quirky, and so we tend to hire the people that like to work in this culture. And so that's a little bit self-selecting. I'd say, you know, we're super transparent internally. I just told you the 2 programs we're working on, for instance, and like everything about the platform. Like we open source the back end of our platform to release a COVID test, right? So we are culturally, in that sense, self-selecting, I guess.
And it sounds like a great selection. It is. Having this team, having set up your lab in the Bay Area, I'd love to hear a few of the milestones or accomplishments that you're most proud of.
I think, you know, like one of the funny things is like, yeah, we released this COVID test. It was like, it's kind of like the back end of our high throughput platform. We can measure many RNA barcodes at the same time across tens of thousands of wells. We built that to be really cheap. We had optimized it over a couple of years, and we knew that would work. And, you know, it's just RNA, small pieces of RNA. So like, it's like we could probably like amplify parts of COVID and test that too, right? And parts of the human genome. So So we did that and it worked really quickly. We open sourced it first on Twitter, then on like the open COVID license. A bunch of companies started taking it up. I think, like Guardant Health and Helix got EUAs on the basic Ginkgo Bioworks. And we kind of launched a facility at UCLA. So it was just like, it was cool to see like a very like straightforward technology for us. People thought you couldn't use sequencing really as redone. Like if you look at like UCLA today, like they've done like 1.5 million tests, and like across the campus, there's like vending machines with the technology, right? So like, I think that stuff like that, I'm really proud of. Like, hey, we've actually helped a ton of people. And that to me has been a lot more gratifying than almost anything, like anything I've ever published in my career, for instance. So we do like building things that matter. I think obviously we've made a lot of progress. Like we built a whole high throughput synthetic chemistry capability. We kind of really like, we run, probably 10 to 20,000 novel compounds a week across like multiplex assays, like week in, week out. Like we did 100 platform runs last year. So like really turning a science project into an engineering project has been really gratifying. And then like, we've actually seen proof points. Like we can actually engineer polypharmacology and bias in novel ways. We can actually measure those in, animal models at this point, both like mouse and canine models at this point, but like for our furthest along programs. But like, I think what I'm most proud of is that we're like able to take these kind of complexities, boil them down to mechanisms, and then build molecules to modulate that and then go and test them, right? And we're not in humans yet. We hope this, funding will take us towards that. And I'm really excited we just closed the Series B, and I'm excited for the opportunity to be able to do that, and even in, what is tough economic environment.
And a phenomenal answer it is. I had no idea about the vending machines. That alone is a great demonstration of the technology and the fact, as you said, that you're thinking a little bit outside the box, which is always exciting. I love the engineering approach, coming from an engineering background myself. But
yeah, no, thanks. I mean, it's been new to me in some ways, but it's kind of hiring a different type of people in some ways that has brought that to us in the past couple of years. So we're yeah, it's been awesome to kind of see them build the platform and see the scale where we thought we could get to someday, but like it's here and it's working, and that's cool to see.
Well, it's here in 2 to 3 years as well, which must have been incredible, the pace that you're moving at. We'd be interested in then pivoting a little bit to chat about maybe the next 2 to 3 years or moving forward in the future. So what's coming next for Octant?
Yeah, so we want to advance our 2 current programs towards the clinic. That's one. We just raised this amount of money. The other things we want to do is we want to like really turn the crank on one of those kind of hypotheses, and in particular the correctors hypotheses. We think there's like a bunch of rare diseases that we can go after next, where we're just like literally repeating the same thing. We have the reporters, we have the thing set up. We can literally just run the chemistry campaign through this high throughput chemistry system. Just to give you an example, like the rhodopsin program, we probably built like, I think on order north of 200,000 custom compounds for that program, and screen it against like these many mutations, right? Like, so I think that's been, and seeing the results of that, like we just want to turn the crank on maybe 5 or more of those kinds of indications. In order to do that, we basically have to scale the platform. So bringing in a lot more of kind of walk away automation and really build out the software and data stack to be able to handle that is going to be it's going to be challenging. But I think those are the main things. Is like we kind of have this platform 2.0, which is basically scaling what we have, like turning the crank on a bunch of indications in the correctors, advancing our current programs in the clinic. And there's a few skunk work projects that I won't talk about.
Absolutely. You mentioned like a few areas that you're hoping to kind of expand on, say like in the automation or walk away automation, to be able to scale up. We're curious if there are any other technologies that you would be really interested to see improved or innovated or invented that could help Octant in what you're aiming to do.
Yeah, so I'd say one we're doing is like this that I didn't talk about yet, is we did we're really doubling down on this deep mutational scanning capability. So I had mentioned that we had used it for like kind of not like the human genetics and some of the correctors work, but one of the things we're really excited about is using that type of work for structure activity relationships as well. This idea to like how you modulate the particular protein and how it affects different pathways is really important. And like you can use mutation to start doing a lot of those things. So our partnership with Bristol Myers Squibb that we announced recently as well, there we're working on a class of receptors and in a set of immune conditions. So then built out a team to really build out the engineering culture of that process and apply it to important targets. So that's one thing that we're really excited about. And a couple other things internally. I think more broadly, I think is the question you were saying, which is what other technologies do we intend to bring or are getting exciting? I am getting excited about, like, we internally produce a ton of data on how chemicals interact with targets of human physiology. I'd say our data is pretty world-class and mechanistic in ways that are interesting. And so bringing more computational approaches in relating chemical structure to those kinds of activities around ML and AI, and like, how do we turn that platform? And so I'm getting excited. Like, I wasn't excited for the last few years because I didn't think the data was there, but the data is definitely here now. And so really building out that data infrastructure is something I'm super psyched about. I think more broadly, you know, I'm excited by a lot of the innovations in companies that have been started over the last several years. Like, I think there's a lot of really interesting approaches. I am very excited about molecular kind of glues, these kind of, or what are, you know, induced proximity systems, things like PROTACs. Like, how, you know, I think there's a lot of logic in the cell that happens through proximity. And if you can use chemistry to do that, it opens up a whole class of really interesting kind of ways to alter physiology that didn't exist before. And I think we have like a killer platform to do that. And I'm kind of itching at the opportunity to start applying it to that. But, you know, I think the challenge of any startup is trying to stay focused as much as possible. And so I would love to see a lot of that kind of start playing out as well.
And I know we would also. I'm personally incredibly curious to learn more about the high throughput synthetic chemistry platform you talked about. But before we come to a close today, a few rapid fire questions.
Yeah.
So imagine it's 2050. Can you give us a sense of where the field of treating complex diseases has reached, and where is Octant in that space?
I am consistently always stunned about the complexity of biology and how far we are. Like, we're like prodding an alien, like, basically at this point. So I still think we have a long way to go. Maybe the answer, Octant, where I would like to see Octant in 2050 is like, like, I love the idea of leveraging the 7 billion people on the planet and its genetic variation to understand how modulating targets impacts phenotype. I think that's doable. And I'd like Octant to be there. Like, how do we, like, every single mutation compatible with human life exists in the diaspora, and like, can we interpret that through the lens of biochemical mechanism to understand like how impacting biochemical mechanism impacts phenotype? I think there's no reason why we can't do that. So like, I'm very excited that Octant would be at a place where we're successful enough that we can try such a large experiment. I think the other side of that is like, ultimately, this interaction between chemistry and biology, I think, is so fascinating. And like, understanding how chemicals interact with the targets of human physiology, these proteins, there's only 20,000 of them. There's more cells than that in each well of a 384 well plate, right? Like, at its most manifest, can you really look at how a chemical interacts with the target's physiology broadly and use that to build compositional and more rational ways of building small molecules that achieve biochemical functions that you want them to achieve? I think it's small molecules are in for a renaissance. And I think a lot of the things we think can be only done by gene therapy and other things are going to be done by small molecules. And so I'm super excited about that.
And I think we're all excited to see it play out. Pivoting slightly, do you have advice you'd really like to share with grad students, professors who are interested in starting a company?
Yeah, so I talked to a lot of professors and students over the years. I mean, nothing other than like life is short and you should do what you like, manifest yourself into the world. And if that's through academia or it's through industry or whatever you want to be doing, there's a lot of opportunity right now, and that's exciting, right? And so take advantage of it, I think, more than anything else.
A strong message. And do you have any other closing thoughts, shameless plugs, anything to share with the audience?
Yeah, well, thanks for doing this. B, we're hiring quite a bit. So I think we have like 25 positions open on the website. I'd say in terms of like closing thoughts, yeah, I mean, you probably know of like the biotech plunge over the last 6 months, but I am incredibly excited about like the teams that are coming together, Octant being one of them, but like I just see a lot of stuff in the space. And I think it's an exciting time to be in biology and healthcare, like not just like therapeutics, right? Like just I think like a big digital transformation in our ability to like deliver healthcare better. I think there's no more, I think if COVID has shown anything over the last several years is that A is like, it's super important. Like when we lose our health, it's like nothing else matters. And then B, like, I don't think you would have picked Moderna and like BioNTech to win the vaccine war and how much that changed the world, right? Like new technologies, they're worth it. Like you can think about like how, if the allocation of our like capital is related to like the importance of the problem, then I think we're still vastly underinvested in human health. And so like that there are good bets being taken, and I'm excited, and I hope that they continue to be taken over the course of time. So yeah.
A message I think we're in incredible agreement with. It's something that really resonates as we see this rise of tech bio companies like Octant and others, and we see the impact that investing in health and as well as wellness can really have on society and the world at large. So thank you for sharing that message with us, as well as more about yourself and Octant today, and for an absolutely incredible episode, Sri. We're very grateful for your time. Thank you again.
Yeah, thanks for having me on.